Okay, so I'll get started then. Well, I hope everyone's doing well. My name's Donna. I'm an EngSci 2T2, and today I'm going to be talking about some really cool research I've been doing this summer. And we looked at astrocyte to neuron conversion in the medial prefrontal cortex. Don't worry if none of those words make any sense. I'm going to go over it in the background information. So most people are familiar with what stroke is. It causes injury to the brain and is actually one of the leading causes of adult disability worldwide. So we looked at an area of the brain called the medial prefrontal cortex. And this is an area of the brain that's responsible for executive functioning and problem solving. So when you have stroke here, you're going to have a lot of cognitive deficits. So in my title, I mentioned two types of cells, neurons and astrocytes. And these are actually the only cells you need to know to understand this research. So neurons are a type of cell in the brain, and they're responsible for transmitting information throughout the central nervous system. And during stroke, neurons actually die off, and that's why we see the deficits that we do associated with stroke. Astrocytes are another type of cell in the brain, and the main thing we need to know about astrocytes is that in response to injury, astrocytes become activated, and they actually congregate around the site of injury and form this sort of dense physical barrier. Another thing about astrocytes is they express markers such as GFAP. This is just going to be important because this is how we're going to identify and target the astrocytes later on. So I just mentioned stroke obviously is a horrible thing, and it depletes the neurons in the brain. So we wanted to come up with a method to promote stroke recovery, and we thought of cellular reprogramming. So cellular reprogramming is just the conversion of one cell type to another. And I mentioned astrocytes and I mentioned neurons. And previous literature has actually shown that you can actually change astrocytes into neurons by using the transcription factor NeuroD1, and you can do this in vivo. And our lab has actually shown this exact method in the motor cortex, which is another area of the brain, and we have shown that this conversion does work and it does enhance functional recovery following motor cortex stroke. So now we want to do the same thing, but we want to do it in the medial prefrontal cortex. So we want to use direct cellular reprogramming, and we want to try and see if the cognitive deficits caused by stroke can be restored. And underneath this overarching goal, we have two main objectives. So we want to examine the reprogramming efficiency. So how many cells, how many astrocytes can we actually turn into neurons? And we also want to assess the impact of reprogramming on behavioral outcomes. So I'm going to talk about the reprogramming efficiency just because that's what I worked on the most this summer. And our hypothesis with regards to this is that the expression of NeuroD1 in astrocytes will result in their conversion to neurons. So in terms of how we're actually doing this, we're using something called an adeno-associated virus, and AAV5 in particular. And essentially what this virus does is it's going to deliver NeuroD1 to astrocytes under the control of a GFAP promoter. So remember how I mentioned that astrocytes express GFAP. So this virus is going to get into cells, but only if the cell expresses GFAP is it then going to be able to deliver its vector, which includes NeuroD1, to the cell. And then once this has been successfully delivered and expressed, the cell is going to be marked with a permanent fluorescent protein that lights up red, and that's how we're going to know that the cell has been transduced by the virus. So on the bottom here, I have a little timeline. So we're giving the virus 7 days post-stroke, and then we're looking at various time points after to see what's happening. So we have 3 experimental groups. We have the sham Cree, so this is a healthy mouse, and we're injecting it with a control virus that doesn't have NeuroD1. Then we have the stroke Cree, so this is a mouse that does have stroke in the medial prefrontal cortex, and we're still just giving it a control virus that doesn't have NeuroD1. So remember, these 2 groups are the controls, and we are not giving them NeuroD1. This is important to remember when we discuss our results. So no NeuroD1 here. And then in the experimental group, we have a mouse that does have stroke in the medial prefrontal cortex, and we're giving it a virus that does have NeuroD1. In terms of how we're actually counting the reprogrammed cells, we're going to be using immunohistochemistry protocols. So here's a very pretty image of some cells in a brain. And all the cell nuclei are going to be stained with DAPI, which shows up blue. All cells transduced by the virus are going to show up red, like I mentioned earlier. And then all cells that are neurons are going to show up green. And if a cell has both been transduced by the virus and is a neuron, we're calling these the reprogrammed neurons. And what we assumed happened is the virus got into an astrocyte and the astrocyte turned into a neuron. So we're looking at the 3 groups, 45 days post-stroke or 38 days post-reprogramming. And here's what we expected. So on the y-axis, we have the percent of reprogrammed neurons over total transfected cells. And we're expecting for the control groups to have lower reprogramming efficiencies, just because, remember, we aren't giving them any neuro D1, so there's no reason why we should see astrocytes turning into neurons. Then we expect a higher efficiency in the actual experimental group. So we actually did this experiment in the motor cortex, remember, and we did observe what we expected in the motor cortex. So as you can see, the two control groups, we're looking at like 3%, 6% efficiencies, very low. And then in the actual experimental group, we're seeing like 20%, which is significantly higher. But here's what we saw in the medial prefrontal cortex. Yeah, and you can immediately see that this is completely different from what we both observed in the motor cortex and what we expected. We're seeing similar quote-unquote reprogramming efficiencies for all three groups, and just looking at even the first control group, we're looking at an efficiency of above 60%, which is crazy. So immediately what we wanted to do was we wanted to compare the motor cortex and the medial prefrontal cortex. So we took the sham Cree, so this blue bar, and we compared the two. So we looked at 5 days post-injection and 14 days post-injection, and we just looked at what the proportion of cells transfected by the virus were doing along these time points. And just focusing on the green bar here, so this green bar represents the proportion of neurons, you can see that the total the neurons aren't really changing in the motor cortex. The proportion isn't changing 5 days to 14 days. But in the medial prefrontal cortex, you can see that the relative percent of neurons expressing the virus changes significantly. So a proportion can either increase because the neurons themselves are increasing or because other cells are dying off. So we quickly looked at the raw numbers, and we saw that just looking at this green bar here, the number of neurons themselves does grow sharply from 5 days to 14 days post-injection. Okay, so just to quickly summarize, so we're seeing something completely different in the medial prefrontal cortex, and it seems to have something to do with our raw neuron numbers. So we're seeing just a lot more neurons being transfected by this virus. So what is happening? Why are we seeing these reprogrammed neurons when we aren't even giving them the transcription factor? So we have two hypotheses. There's actually another type of cell in the brain that expresses GFAP, so the virus can actually get into these. And these are neural precursor cells, and they just become neurons without neuro-D1. So we were thinking maybe the medial prefrontal cortex has these cells in them, and we just weren't aware of that. So that's one option. And then the second option is that the GFAP expression itself is different in the neurons of the medial prefrontal cortex. Maybe the neurons in the medial prefrontal cortex express low levels of GFAP, and that's why the virus is getting in. So in terms of next steps, we want to use RNA-scope to determine if the medial prefrontal cortex neurons do express low levels of GFAP. So that's our preliminary next step, I guess. We still have a lot to do in terms of understanding what is happening in the medial prefrontal cortex. But just to summarize, overall, our findings have proven that reprogramming does have potential as a therapeutic to promote stroke recovery. And I genuinely think this idea is so cool because we're taking cells that are already in our brain and just converting them into the cells that we need. And I genuinely think that if this could work, it could completely change how we go about treating brain injury. So thank you for listening. I just want to put up my acknowledgement slide just because I couldn't have done this without everybody at the lab, and they were so helpful to me. So yeah, I'm open to any questions now, and thank you so much for listening.
Hello, Donna. This is Sharianne speaking. How are you?
I'm great, thank you.
Thank you for such a very well explained presentation because I know nothing about this and I was able to understand. So thank you for that. I was just wondering, so with respect to the experiments being done with mice, how far do you think you are with respect to having the experiments done with a human participant? And my second question is, are you at all looking at therapy along with the reprogramming of the neurons, or is it just reprogramming what you're looking at?
Okay, well, in terms of the first question, that's actually a really good question. This is obviously still very far into the future just because we haven't figured out what's happening in the mice yet, but we actually want to use cerebral organoids which are essentially just, it essentially allows us to test this method on human cells without actually using human patients, just in case something goes wrong. So in terms of testing it in the future in humans, that's our next step. And then I think you asked in terms of therapy and using this reprogramming method. I definitely think that by using some form of therapy or rehabilitation in conjunction with this reprogramming could definitely be of benefit. Current stroke treatments actually do rely heavily on rehabilitation, and I definitely think that we can look at it with this method as well. So hopefully that answered your question.
Yes, thank you very much. I know with research like this, it's difficult to put timelines on it. I just thought I would ask to see if there was something broad that would.
That was a really good question. Thank you.
Are there any more questions from the judges? Okay, if not,
Well,
thank
I
you.
think Simon raised his hand.
Oh, I'm sorry. Yep, you can unmute.
Oh, I don't think I can hear anything.
Hello? Hello? Can you hear me?
I can hear you, yeah. I can't hear the question, though, I don't think.
Well, maybe while Simon is working on maybe the microphone, just a quick question, Donna. Yeah, I found it very exciting work, and thank you for explaining it so clearly. I wonder, do the surprising results lead you to the conclusion that ND1 is not as effective, or is it more that there are many more mechanisms that need to be understood before talking about ND1 or any other method of this reprogramming?
That's actually a really good question. So obviously, when we saw these results, our first instinct was, okay, what went wrong? We actually, personally, I don't really think that it's the NeuroD1 itself that has gone wrong, just because we have seen this in the motor cortex to work, and we have seen it. Other papers have done this in other injuries, such as Alzheimer's, stab injuries, and it still worked. So I genuinely believe that it's something to do with the environment of the medial prefrontal cortex itself, which is why most of our hypotheses right now are, I guess, assuming that the ND1 has worked perfectly, and we are sort of trying to investigate the environment of the medial prefrontal cortex itself. So yeah, I think that's where we stand in terms of what's happening.
So it seems like your research is highlighting how different this medial prefrontal cortex is.
Yes. Yes.
Okay, thank you.
Thank you for the question.
It looks like Simon has written his question now.
Oh, perfect. I'll read it then. Do you think that the virus itself can induce the proliferation of neurons by integration at a specific site in the genome? Actually, this is a very interesting question. I personally, I don't know a lot itself about the mechanisms of how the virus impacts the genome itself, but this might be of interest to you. We actually just read a paper in our lab that showed that after stroke, astrocytes can actually, in the striatum, they can actually become these sort of like neural precursor cells, and they can actually give rise to neurons. So that was actually something that we discussed. We were like, okay, maybe the virus itself is changing the astrocytes, and it's not even the NeuroD1 that's doing that. And so in terms of can the virus itself induce the proliferation of neurons, that's definitely something we considered. It's just currently I don't believe there's a lot of research or papers on this information. So we're currently sticking with trying to analyze the environment itself before we jump to like, oh, maybe it's the virus that's changing the astrocytes and making them into neurons without already one. But that was a good question. Thank you.
All right. So if there are not any more questions, thank you, Donna. And next, I would like to